Dr. Nathan T. Mortimer. He makes fruit flies do superhero level science, y'all. From Oregon State University, a molecular biologist. His lab explores how cells respond to invaders, stress, parasitic wasps. Yep, he uses fruit flies to study wasp venom and how our immune systems can make decisions on processes that go wrong. He can also, this is one of my favorite parts, use wasp venoms to actually modify them and make them for drugs and treat ailments that otherwise didn't have any treatments before. Not only, though, is Nate an incredible powerhouse researcher, he's a dedicated mentor and educator. He taught me everything I know and Lita know about how to think and act like a scientist. He leads a national undergraduate genomics program and helps tons of students yearly across many institutions get hands-on work in real science. So he investigates the immune pathways, he builds the next generation of scientists, he brings passion, energy, and love of curiosity to everything he does. Guys, please welcome one of my most best friends, Dr. Nathan T. Mortimer.
Awesome. Thanks, Balint. Thanks for having me and everyone for being here. Hopefully you'll write my promotion letter for me.
I am absolutely happy to do that, Nate. You know that. Would you mind, Nate, giving us a brief introduction on who you are, and then of course this part will be a little bit longer, but like what got you into science? Like what was it that attracted you towards the sciences and made you want to study something in a lab?
Yeah, no, that's a great question. So I was born in Canada, in Ontario, and grew up there until I got into high school, and then we moved down to the States. I did all of my education at Emory University in Atlanta, which was a fantastic place to live, but I'm glad to be out of it now, out of the heat. Since then, I've had independent faculty jobs at Warwick University in England, also Illinois State University before moving to Oregon State University a couple of years ago. And we really like it here, really happy in Oregon. It's a great place to be. I really got started. So I don't know if there's any other Canadians in the audience today.
We've got a lot of and actually happy belated Canada Day, Nate.
Yeah, great. Thank you. So hopefully this resonates with some of the other Canadians. I got interested in science from watching The Science of Things on national TV every Sunday night. Hosted at the time by David Suzuki. I'm not sure if he's still doing it, but that was what got little Nate excited about science and really interested in it. And from there, it became something that was almost a hobby, was reading about science and learning about science. And then, when the time came to go off and get an education, I liked science, so everyone told me, oh, you should go be a doctor. And so I thought I was going to go be a doctor. And then I started working in a lab when I was an undergraduate, and I realized that was what I should do and not be a doctor. So we kind of changed midstream and went, I worked in a lab for several years before starting graduate school, and then eventually went the PhD path. And here I am now with my own lab and having a ton of fun. And the thing that's really cool that I imagine we'll talk about if people have questions about, but one of the big projects my lab has worked on is this ability to do self-recognition by innate immune system. And we do it, of course, in flies. And the mutation that we use to figure it all out was actually originally discovered by David Suzuki before he was a TV presenter and when he had his own lab. And so there's something really sort of full circle about working on something that my boyhood hero had discovered. So that was a very cool thing to do.
That is really, really cool. And it's beautiful about that whole full circle, right? Like as a kid, it's like, oh, this is what excites me about science because it's this guy's TV program. But it was his mutation that got you excited and studying it. Did you, when you found this fly that you were interested in studying, what came first, your knowledge that it was the same gentleman who did the hosting? Or was it just like, oh, this is a cool mutation. Oh, by the way, there's this other effect.
It was totally driven by the mutation, right? So I was really interested in trying to figure out how this whole process worked. I found a mutation in an old paper that no one had worked on for decades, and it sounded like exactly the thing I wanted to figure out. And so I got into it and I got the flies and I started playing with them. And you were there at the time, and we were playing with these flies and trying to figure out what they were doing. And then one day it dawned on me, like, oh, well, David Suzuki used to do this. And so I looked it up, and he was the Suzuki that the flies are named after, which is really funny.
That's so cool. I didn't realize that there was a gap in that time from it. Like, I thought it was like, oh, like we got the flies, and then it was just like, oh, there was like, yeah, this is the show guy. But like, it's even cooler that there was a long while to it.
Yeah, it took me like at least a year to figure it out.
So Nate, how did you? So like after the graduate school, I guess your undergrad, how did you decide, and why did you decide fruit flies were something that you were interested in? Right, a lot of questions that we will get sometimes is like, well, why do I care about fruit flies, and like why are fruit flies interesting? So not just from like there's a human perspective of it too, but also just like a personal interest of it. Like what was it that drove you? Was it the organism? What was it? The questions that you could ask? Was it both? Like, what drove you guys?
Yeah, no, that's a great question. So when I started doing research, it was an entirely different thing. So I started doing pharmacology and behavior with rats. Was my starting point. And I loved it. It got me really invested in the lab. But I very quickly realized that my interests were much more at the molecular level and even at the biochemical level. And so I was a little bit frustrated, is not quite the word, but frustrated working with rats or mice or something that, has a long lifespan. You have to plan all of your experiments well in advance so you can get approval for them. And then that was when I was an undergrad. That's what I did. And then when I was looking after I graduated, and I was looking for a job, and I got this job offer to work in a lab that did fruit fly research. And I kind of thought going in like, well, that's a bit of a weird thing to study, but I need a job, so I'll see what it's all about. And I went in and I fell in love really quickly with it. The biggest thing to me is we have so many tools available in flies, right? So the fruit fly genome has 13,000 genes. Almost all of those we can knock out on demand in any tissue we want. A good chunk of those we can overexpress in any cell type at any time we want. And so it's amazing the level of sort of mechanistic detail that you can go after. And in such an easy way, right? Like working with flies is not hard. It doesn't require, I can train someone to do it in an afternoon, and then they can go on and do this really, really advanced analysis of how proteins interact with each other, or how a signaling pathway dictates the outcome of an infection event, right? Like we can get very, very detailed information in a way that's either not possible or would take a really long time in a lot of the other model organisms. And it's great that they are very easy to translate back to human research, right? So flies have versions of 70+% of all the genes are known to cause disease, and they do very similar things in flies to what they do in people, right? So we have fly models of all different cancers, neurodegenerative diseases, heart diseases, and we can manipulate essentially the same protein in the fly and learn a lot about it, learn how to turn it on, how to turn it off, how to manipulate it with a venom protein that I'm sure we're going to talk about. And so there's an awful lot we can learn, and then it translates back fairly easily to thinking about how do we use this in a practical sense for people who are having a disease. Or kind of one of the things that's really emerging is this idea of insect vector disease, right? So mosquitoes vector all sorts of parasites, viral infections to people. And we don't understand their immune responses super well. But if we could, like, how great would it be to cure malaria by making sure mosquitoes never pick up the parasite in the first place?
Right.
That seems way better than someone getting malaria and then giving them a drug.
Or even wiping out all mosquitoes, right? Which could cause all kinds of ecological impacts.
Yeah, exactly. Like you can kind of go in a very directed way, block the event that leads to disease without, like burning down the jungle to get rid of malaria kind of thing, right? You can actually specifically go after, make those mosquitoes immune, right? All of a sudden malaria is done, and with very little impact outside.
Absolutely. Yeah. So with that, you would say like if you understand this model organism or this fruit fly, that we have all these genetic tools, and if you understand how its immune system works, a lot of that is directly translatable to the mosquito. So they're not so different that they would have something like a totally different genes that govern the immunity. It would be very, very similar composition.
Yeah, exactly. So a lot of the immune genes, immune pathways that we figured out in flies are already being targeted in mosquitoes to understand how their immune response works. And I would love to get a little bit deeper into looking at that in my lab. Particularly, we study wasps, and I love my parasites. I'm never going to give that up, but I think it would be cool to get a little bit more into some of the virus research as well, especially since that's such a big pressure coming from mosquitoes in particular, but other insects can also vector viruses. So I think that would be very cool to start understanding antiviral immunity a little bit better as well.
Presumably you could go from other types of diseases too, right? You could go from this malaria, but what about Zika and dengue, right? You could use this for all kinds of different models. A question from chat, and I feel like this is one in that framework too. Can you infect a fruit fly to host malaria and or any of these other pathogens that might be in the mosquito, or do you have to have some kind of surrogate for it?
Yeah, so that's a really great question. And it seems like for the most part, we would have to use an analogous pathogen, not the same pathogen. So something very cool about malaria, the genus of parasite is Plasmodium. And something that's very cool about the Plasmodia is they are ultra specific for their vector species, right? So something that I learned fairly recently, embarrassingly, is that when we think about mosquitoes and you go out and get a mosquito bite, there's dozens and dozens of species of mosquitoes around the US. In Atlanta, there's a ridiculous number of mosquitoes, right? And only certain ones can vector certain diseases, right? And that's true among Plasmodia, right? So there is 2 or 3 species that can cause malaria in people. There's also several species that cause avian malaria. And so those parasites can't actually cross either the host species boundary, but also not the vector species boundary. So they're very tightly only able to infect one species of mosquito. And so I don't think it's going to be possible for us to take those and directly study them in flies. Mm-hmm. I think what we're going to have to do is either find some kind of organism that we could use as a proxy, or what I'm kind of drawn towards is trying to almost make a genetic proxy, right? So if we can figure out potential genes that are involved in, let's say, how Plasmodium blocks mosquito immune response, right? We could then take and we could individually express those proteins in flies and look to see, can the fly immune response recognize it? How does it recognize it? Can we manipulate the immune response so that it now does recognize it, right? So kind of making almost genetic models. It would obviously take a lot more work kind of going one by one through it. But I think that might be the way to get around a lot of these potential issues that we face thinking about crossing one species to another.
Yes, too, by that, the one at a time that you're going through, while, like you said, it would be pretty tedious, but you'd actually figure out what the recognition is, between the host and the pathogen, right, on the molecule, not just there's recognition, which you would predict, right? Because it's an animal inside of you, but okay, it's this is what we need to focus on, this one very, very focused piece of it. And that's just would be so cool to be able to do.
Yeah. And you're exactly right. It would be a big undertaking, but it takes us from 0 to 60 really fast once we find the gene or whatever it is that's responsible. It gives us a lot of information right away.
And because we have all the genetic tools already there in the fly, we could then start we're at 60, but now we can go to 100 pretty quickly too, right?
Exactly. You can then figure it all out very, very easily.
A question from the chat, and this comes up a fair bit too, is when we're trying to fight one of these ecological diseases, if we're trying to cure that mosquito of malaria, is there a desire to eliminate it? Is there some kind of ecological benefit to malaria or these Plasmodia? When we're thinking about getting rid of it, do we anticipate anticipate there being some kind of big impact?
Yeah. No, I love that question of thinking about the consequence of trying to wipe out some of these parasites. It certainly is a question to be answered by someone far smarter than me. It's something that is important to consider, right? So is there some kind of benefit to having these what we view as disease-causing parasites in the ecosystem? There are a whole family of parasites that are related to malaria, related to Plasmodium, that are not transmissible to people, and that infect a lot of different mosquito species that malaria-causing parasites don't infect. And so it seems as though it is a really rich ecological niche for these parasites to live in. And so it would be, I think, really fascinating for someone to look into kind of an ecological impact of Plasmodium infecting mosquito, and how does that change mosquito population, mosquito behaviors. I think a big push obviously is to try to eliminate malaria to prevent burden to human health. And causes death, obviously, is a worthy goal to look at. But we do need to think about those sort of ecological outcomes, right? Because you kind of mentioned people have had these ideas of, well, if we kill all the mosquitoes, then there's no malaria. But that doesn't seem great. It seems like the mosquitoes might be an important part of systems that are existing in these places.
It almost seems like a smidge of a backwards question almost, right? Because it's like, well, why would you? Of course you'd want to get rid of it because it's a disease, but at the same time, it's actually really insightful because it's like, well, it's there for a reason, right? It didn't just evolve for the sake of annoying us as humans, right?
And so it was probably around long before us,
right? And so it's just like, it's the same reason why you don't want to get rid of every mosquito either. There's a lot of pollinators. There's all these kinds of other important elements coming on in, and it's just a really, really fun question to be able to ask.
Sure.
Oh, and Ari, thank you for that gifted subscription to Seronx. Thank you, Ari. The gifted subscription from the lovely Ari Summer coming in. So yeah, Octavius, wonderful question. It's one I also love your answer, Nate, that we can't really answer it.
Yeah.
Right? Like, we don't know. There could be a huge effect to it. Downstream and upstream, and I think that's really a good kind of way to answer this kind of question as a scientist. Is like, there isn't just this yes or no. It's like, here, let's actually break down these answers to it. And one of our people, Hot Local Grandpa, says, I just wanted to say that science is incredible and thank you for doing what you do.
Oh yeah, I love it. It's the best job I could imagine. So I'm glad I get the opportunity to do it.
And for just obviously coming on with us today, Nate. I know how busy you are, and like, you're mentoring people, you're bringing, you're teaching people the science, you're bringing up the next generation, you're writing grants, you're writing papers, and now here you're with us, and we just got another 40 people up on in here with another raid.
All right.
So we've got 700 plus people watching here today. Supreme Doom coming on in. She's one of our maker and crafter friends. She's actually from Canada. Raiders coming in. Our guest, Nate, is originally from Canada. We were chatting about Canadian TV shows about science and the exciting component, how that inspired Nate to go down the scientific route, and how the mutation that he studied was actually by this gentleman who did the show. And we got another raid. We got another 88 people coming on in here, Nate. Irish John Games coming in with a raid. We got double rated, folks. Welcome on in, everybody. Raiders coming on in. My name is Balint. I'm 1/4 of the SciAnts stream team. The other 3/4 are my wife, Lita, our daughter, the Munchkin, our brand new son, Buster Brown. Lita and I are research scientists, PhDs in molecular biology, and today we're joined by actually 2 scientists today, celebrating all things science today. We're joined by, right now, Dr. Nathan T. Mortimer, an incredible scientist from Oregon State University, studying venoms, wasps, treating malaria, and beyond. He's also my best friend. He was the best man at my wedding. He knows a bunch of science and Balint stories that maybe would raise an eyebrow or two. But y'all, welcome on in. We're just getting kind of started about why Nate's studying, why it's important, why we study it in the context of how we study it. We're kind of getting into Nate's journey before we get to the hard science, and there's a lot of it to chat about, so please ask your questions. Nate is, again, I learned everything from Nate, so please ask away. That's why we're here to all together learn. And folks, for already here, please go follow our friends Irish John Games and Supreme Doom. I hope you all had a great stream. If there's anything you want to share, feel free to throw it into the chat. We'd love to show it off to the party people after Nate's on with us. So please, again, share all the things. Thank you all so much for entrusting us with your community. Nate, let's I guess continue on. Right,
Yeah.
and chat about the beauty of what you wait. So for those coming on in, Nate was telling us he picked fruit flies. They have all these genetic tools that we can do. There's all this like really amazing genetic tricks that you can do. What question did you study, Nate, as not as undergrad, but in grad school? Like, what got you excited about I guess using fruit flies as an undergrad or from undergrad to grad student, and then how did you transition eventually to wasps?
Yeah, no, that's great. So when I was a graduate student, I started out very sort of cell biology, biochemistry focused. I started working on a protein that is what's called a ubiquitin ligase. So this is a protein that its job is to regulate the levels of other proteins.
Supreme
right?
Doom
So it will
56
say, okay,
just
we need to
subscribed
turn
for
off a
21
particular
months.
signal. So let's find the protein that's turning that signal on and let's just get rid of it, like literally chew it up, degrade it. So that's the job of what this protein does. And so I started studying it and understanding, how it targeted other proteins, what it used, what kinds of proteins it targeted. But what's cool about the fly is it's not just a bag of protein, right? It's actually an organism that develops and has organs of its own, right? And so I was able to look and see not just that this protein that I was working on regulated protein X, but if you change that regulation, it alters the fly itself, right? And so I ended up studying proteins that dictated how flies grow, the organ that they use to transfer oxygen, right? So insects are not quite like us. They don't have a closed circulatory system, but they do have this tubular network called trachea, and that's what carries oxygen all throughout the insect, right? And so it's this sort of passive diffusion that it moves by, but they have these tubes that carry the oxygen. And that's actually why, when you think about sort of the prehistoric insects, they have these great, huge insects. It's because the environment had a lot more oxygen back then. And so it could actually just literally diffuse more oxygen through these tubes. And so the organism could grow bigger. And so our insect size is kind of dictated based on how much oxygen is in the atmosphere, which I think is one of those weird things that I picked up along the way, but I think is really cool.
It's kind of wild, right? And that's like the oxygen composition has changed in our atmosphere today, and that's has influenced the insect size.
Yeah.
It's really, really cool.
Yeah, it's remarkable to think about. And so that's what I ended up studying sort of by mistake because I was biochemically interested in this transcription factor, and that's what its job was. And it really struck me this, I think beauty is the right word, right? That I could understand one mutation in one gene in the genome of a fly, and then I could look at the fly itself and see the outcome of that one mutation, right? We can change one gene and it alters how the fly develops.
So,
right?
So for you, the attraction was that we can go from a very small molecule, like that protein in an animal, like microscopic level, to the macro of the whole animal, and back to it.
I really love that crossing scales, right? You talk to most biologists, right, and you'll hear them say, oh, I'm an ecologist, I study ecosystem, or I'm a biochemist or a biophysicist, and I look at how this electron passes from one atom to another, whatever, they do. I don't know what they do. For me, what I think is really interesting is spanning those, right? Is being able to think about one little change to one little molecule and the impact that has on a whole organism. That just kind of blows my mind. And that's really what got me hooked into studying wasps, right? So wasps as parasites of fruit flies, because we now have this situation where we're looking at an interaction between two different organisms. And I've been able to start learning individual proteins that dictate the outcome of that interaction. right? Like we can make one change to a fly and it completely changes its ability to interact with a whole other organism. And so we kind of understand every level from this molecular scale all the way up to essentially an ecosystem, right? Two organisms interacting with each other. And I just think that is the coolest science that you could possibly do, right? Like I think I would get really bored if I focused in on just one level.
So
I have that ability to ask questions that span different domains.
So Nate, I'm going to rabbit hole us then. We're going to go out of order because, I mean, we're both excited about this now, so let's go down this rabbit hole.
Yeah, let's do it.
So when you're saying the two different organisms, can you give us the scene? Like, what are the two organisms that you're studying?
Yeah.
How does one study them, right? And what questions can we answer before even we start doing the genetic manipulation?
Yeah, no, so that's great. So we can think of it very generally as a host-parasite interact, right? And so essentially what that means is we have a parasite that is going to infect the host. It's going to exploit that host for its own gain. Our organisms that we study, the parasite, are these species of parasitoid wasps. And we can certainly talk about what parasitoid means in a minute. But the idea is, so we have these parasites, these wasp species that infect Drosophila, so fruit flies, which are our hosts. They infect the fly during one of the developmental stages, right? So essentially during the maggot stage, right? So the adult fly will lay an egg, that egg will hatch, and when it hatches, it is in the maggot stage, and that maggot will develop for several days, then it goes through pupation and becomes an adult fly, right? So it's during the teenage years, essentially, is when it gets infected by these wasps. And the infection is kind of brutal, right? So these parasitoid wasps are, of course, closely related to the wasps that we all know, the yellow jackets. And if you've ever had an encounter with a yellow jacket where you got stung and it hurt, you essentially experienced what the flies experience when they get infected by a parasitoid wasp, right? So the stinger that yellow jacket used is actually a structure that's derived from the ovipositor, is what we call it in parasitoid wasps. So it is a long, thin, yet really hard structure that they can use to pierce the skin of their targeted host species. And actually, in the case of parasitoid, they pass an egg through this ovipositor, right? The yellow jacket obviously doesn't use it to lay eggs, so it's not really an ovipositor anymore. But it's that same structure that it was derived from the ovipositor. And then, of course, the pain that you feel is the venom that gets transferred into you when you get that sting. And these parasitoid wasp ovipositors are also connected into the venom gland. And so we have kind of this scenario where the wasp pierces the fly's skin, gets its ovipositor into the cavity of the wasp. It then lays an egg, and it transfers a dose of venom into the host. So it's not too unlike being stung, except they have the added insult to injury of getting an egg laid inside them, which we don't experience. And then essentially the adult wasp cleans off her ovipositor and flies away. And then what you're left with is a fly larva or fly maggot that's been infected. And so inside its body is now this egg of the parasite, and then all of these venom proteins that the parasite injected. And so we've now kind of set up this system where there's a competition between the fly's immune response and the wasp venom protein for who's going to win, right? There can only be one, right? So either the fly is going to kill the wasp and develop as a fly, or the wasp is going to kill the fly and then develop as a wasp, right?
So it's an immune war that you've kicked off there. Like, are you going to beat the immune system, or are you going to lose to it? So it's not predetermined. There could be in either direction that it goes in, depending on the host and the venom. So the venoms aren't necessarily all the same, right? So like one of these wasps might have a different approach than a different wasp species.
Yeah. And so we study, I think, 8 or 10 studies of species of wasps in the lab. And what we're finding that's really cool is they have some things in common, right? So it seems as though all of them have the ability to change their host development, have the ability to change their host metabolism. But then they also have all of these differences that make them unique. And so they, for instance, will try to block the immune response using vastly different mechanisms. Even within the same wasp species, they have backup plans on top of backup plans that they try to use against the host's immune response to make sure that they come out on top in the end.
And this is just for the folks at home. It's the innate immune system, right? So there's no B cells or T cells or anything like that in this animal, correct?
Yeah, exactly. And that's one of the things I really like about studying immunity in Drosophila. So I have an autoimmune disease. I imagine lots of people in the audience do too. So we're very well aware of how our immune systems work, and the importance of understanding how they work and how they don't work sometimes as well. And it's really frustrating to me as someone with a vested interest in understanding immune responses that 95% of all of the research, and I'm making that number up. It's not a real number. It's just sort of my experience of reading the literature and going to conferences. Like 95% of all research done on human immunity is acquired immunity, right? So this ability to produce antibodies, B and T cells. But I think, which is important, right? We know a lot about the world and we can, create vaccines, all these things because of what we've learned. But there is this whole other branch of the immune system, the innate immune system, which does most of the heavy lifting in keeping us protected from pathogens. And likewise can create a lot of the burden when something goes wrong. But not very many people are studying how that works. And I think a lot of it is, well, there's a couple of reasons, right? There's the practical. If you start talking about antibodies, everyone thinks you're really smart and they give you lots of grant money. If you just talk about, there's this cell that does this thing, no one really cares. So there's definitely the sex appeal of studying, antibodies and acquired immune responses. But I think it's also kind of so prevalent in how our immune system works that it becomes really difficult to do good experiments to look at innate immune systems in isolation.
Especially in an animal that has both, right? Because
exactly.
as soon as you stimulate once with the innate, you're going to have feedback from the acquired, right? Or the T cells, B cells, and then what's exactly doing it if you have another infection, right? Is it the innate?
Yeah.
Is it the memory of it, or both?
Yeah. It becomes really hard to know what job the innate immune system is doing in isolation. But it's very important to understand it as well, right? Because a lot of the diseases we have are coming because of dysfunction to innate immune responses. And so it's this kind of cruel circle where they're important to understand, difficult to understand because of everything else that happens when we get an infection. And so the flies are great. They have no acquired immune. And so everything we're looking at is innate. And so we can really start to look at innate immune specific activities, proteins, and get a better understanding of how the innate immune system itself works.
And for the folks at home, how big are these wasps and can they sting you?
That's a great question. They are ridiculously small. So they live their life, or they do their development inside a fruit fly. So they're slightly smaller than a fruit fly. They're ovipositor, while being very robust when it comes to trying to infect a fly larva, would not get far through our skin. So they can try all they like, but they are not going to successfely sting one of us.
So you are not under threat from stinging.
Yeah.
At your job, you're not we're not in fear and working in your lab.
Yeah, exactly. So when I was an undergrad and I was working on rats, I got bit. and one of the things that I wanted in a future experimental system was that it could not hurt me. So the wasps, they even if they get mad, even if they come after me, they just land on me and look kind of cute, and they're furiously working away trying to sting me, but it's never going to happen. So I'm pretty well protected.
From the chat, Nate, thank God it was gonna keep me up at night.
No, we're not like those crazy mosquito people who let the mosquitoes sting them.
Yeah, we've talked about it here. It's like that's one way, right, to feed them, or you can use a mouse, or you can use like a synthetic model. But it's good to hear that you're not under threat from these parasites.
Yeah, no, we're pretty safe in the lab.
So where one thing gets really confusing, Nate, is studying this host-parasite interaction and elements of the innate immunity. How do you do that when, like you said, the venom, it's going to be multimodal, right? There's going to be multiple ways by which that you can fight off the immune response of your host. And like you said, there's backups to the backups.
Yeah.
How does one study that then?
Yeah, no, that's a really good question, and I think the main answer is to start with an observation that leads you to a hypothesis, right? So there is an awful lot going on when one of these interactions. But what we typically do is either, let's say, find a venom protein that we are interested in studying how it works, or some aspect of the immune response that we really want to understand, and start by studying that in isolation, right? So let's say we want to understand, how fly immune cells migrate to the site of infection, right? So we can break down that really, really complicated interaction into all these distinct steps. And so we can say, okay, we want to look at the migration step, or we want to look at the activations. And so we can design experiments where we almost don't care what happens after the point that we're interested in. We can focus in on the specific point that we're in. And something else that we do a lot in the lab to study immune responses is, right, we've been talking about these wasps and all their venoms and all the terrible things they do to the host. But we also have wasp species that can't, right? So something we haven't talked about that I think is actually really cool is that among all of these different wasp species, we said about how they use different approaches to try to infect their hosts. There are also some of them that have very different host species, right? So we have a species of wasp in the lab that naturally is going to infect an entirely different group of flies than the ones that we study, right? And so what that means in a practical sense is that their venoms are adapted to deal with a very different immune profile than what they're seeing when they infect a Drosophila that we study in the lab. And so we essentially have these wasps that we can use to do infections with whose venoms can't do anything to the host.
So it's almost like one of the control experiments, right? Like it's gonna mount an immune response. You're still undergoing the feel of, oh, I just got stung. Like with the larva, you got something attempting to develop in you, but you can fight it off. And so okay, you maybe even do subtractive, right? Like this whole set of things is what goes for the losing column, and maybe this set of things go to the winning column.
Yeah, exactly. You can really start dividing it up when you have these wasps that, are essentially useless against that particular host species.
But in the flip side is, if you were to want to, right, if you had the tools, which is the big if, you could almost reverse the question, right? You could study what the winning method is in this other species of host with that losing wasp that would now win. And then you might have a losing wasp because it's not specialized on that host. So you can, right, the thought process is you go back and forth. That's also like really powerful, like from a thought perspective to be able to do.
So that's still one of the dreams that we're kind of slowly working towards is being able to almost shuffle the venom proteins between wasp species. Like use this wasp that can't successfully infect our flies and add new venoms to it and see which ones now can infect them, or take one that does infect and start knocking out the venoms, and once we lose one that's important, it'll no longer be able to attack. We're making strides, but not quickly when we try to do these things.
So an immediate question that comes, right, wasp venoms. How on earth do you know what a wasp venom is? Like you said, there's multiple things in there must be, right? Because if they do different things, what does that mean? How do you know what's in there? How do you test them? How do you get it out of the water? Like all these like basic questions come into play of like how are you going to work with something like that? What do we need to do to be able to work with it?
Yeah, no, that's really good. So the probably a good place to start is thinking about how to actually get your hands on them to work with them. And so we do that in two different ways, right? So the typical way is to actually dissect the gland, the organ out of the wasp that holds all of the venom.
So is that organ, just for us in the crowd, is that where the wasp, they make it and it's stored in a bag almost? Or like,
Yeah, exactly. So they have a gland that's imaginatively called the venom gland that produces all of the venom proteins, and then it has essentially a giant muscularized bag, for lack of a better term, called the venom reservoir. And so all of these venoms get produced in the gland, get secreted out of those cells, and then just sort of hang out in this reservoir.
And so that's what you need to get, right? Is that reservoir,
exactly,
that
yes.
bag.
You can pull those out, and then you can essentially literally rip it open, and then there's all the venom.
And it just floats out.
Yeah, we have a few steps in the lab to separate the venom away from the cells. But yeah, once you get good at doing the dissection part, you can make yourself a batch of venom in less than an hour, and you have enough venom to do lots of experiments with.
So by batch of venom, do you just mean like if I rip a bunch of venom glands in a little well, and a dissecting well, I'm just going to fill it up with those components and everything's in there and it's going to be stable and I can just mess with it. It's really cool.
It's really neat. So you can get a bunch, and then once you have it, whatever you want to do, you can do, right? So we do experiments where you just take that whole venom and put it on cells and see what happens to the cell. Does the cell die? Does it lose adhesion? Does it stop dividing? We do other experiments when we want to investigate kind of the components of the venom, right? So you can take that venom and run it through an instrument called a mass spectrometer, which is going to let you identify all of the proteins that are present in the venom. So it will separate them all based on their size, and then it will be able to really accurately measure that size. And by knowing all of the genes that a wasp has, we can figure out which ones have a protein that's found in the venom. So that's how we've been able to figure out, the wasp has 14,000 genes and it has 200 venom proteins. We've been able to figure out, in this way, we've been able to figure out what those proteins are.
So you can almost backtrack. Like, I have the genome, but I don't know what genes are involved in the venom, so I'm going to get the venom gland and figure out the proteins and work backwards of this venom protein must have been coded by this gene.
Yeah, exactly.
So that allows you, right, to figure out maybe what piece of the venom cocktail is doing what.
Yes, exactly.
From the chat, and I don't know if this is like a technical approach that you could do, but is there a way to almost make an anti-venom for a particular part of the venom? So like if you took the whole venom gland and that juice in there and you put it onto cells, can you block the activity of one of those components that you're really interested in and see its effect? Do you do that, or do you go in the other direction where it's like, I now want to synthesize or make that one component, put that one component on cells and see what happens? Or is it both?
Yeah, it's both. So depending on the protein that you're looking at, you can do it like one or both of those ways. So a lot of these venom proteins have predictable function, right? So some of them act as, like, one we've studied a lot is an ion pump, right? So it's a protein that sits in the membrane of the cell and moves calcium ions from one side of the membrane to the other. Is a very simple task that it does, and there's lots of proteins that do this. And so people have developed a drug that you can put on that blocks these pumps from working. And so we can take our whole venom, we can add this drug, and then we've blocked just one protein within the venom, and we can see the impact of just that one protein.
So you can figure out, like, is this, critical in the initial stage of almost like colonizing the host, right? Versus downstream, like how necessary is it? That is a really, really powerful tool. That's so cool to be able to, because then, like you said, you do the opposite, where you just synthesize that one pump.
Exactly, yeah.
Yeah. One thing, Nate, before we continue, you said predictive function of the protein. So I guess what does that mean for folks at home? So like when you're looking at these sequences, does that mean you don't inherently know right away what it does? And then how do you, I guess, even guess what it does?
Yeah, no, that's a great question. So the way we start doing that is by looking at the sequence of the protein, right? So by sequencing the genome of the wasp, by identifying all of the proteins in the venom, we can then predict all of the sequences of those proteins that are found in the venom. And so something that's really cool looking across between different species across different proteins is that proteins that do similar jobs tend to have regions that look very similar to each, to the point that there are computer programs that have been made where the program has a database that has, oh, this is what a kinase looks like, this is what a whatever other type of enzyme looks like, and you can run your set of proteins through this algorithm, and it will spit out, oh, protein 1 looks like an ion pump. Protein 2 looks like a whatever kinase, right? So you can kind of use that to make these predictions. And of course, they are only predictions, right? So it's using the whole sequence compared to what it thinks it's supposed to do. Something that we're finding really cool with the venoms is that a lot of them don't do what they're predicted to do. A lot of them actually do the opposite of what they're predicted to do.
Okay, that's actually what I was going to ask, was how do you, like, how good is that prediction? But if it's the opposite, that's weird. Why is it the opposite, and what does that mean for the data set? Does that mean it's a bad data set? Like, what's the implication here?
So that's actually showing us, I think, how good the predictions are.
Okay.
Right. So basically, it's able to look at the protein and say, okay, this protein should be involved in process X, right? And it does that extremely well. What's interesting about it is a lot of these, we call them domains, the parts of the protein that look similar across different proteins. A lot of these domains are very dependent on a small number of amino acids within them, right? So a protein is a string of amino acids, often hundreds of amino acids. These domains even are sometimes tens to hundreds of amino acids. There's one or two that you can change that change the function, right? So you have, for instance, a protease, right? So a protein whose job it is to degrade other proteins, right? And I was just looking at one of these with my students yesterday, 2 days ago, and the domain is like 150 amino acids long, right? So that's quite a large chunk of our protein.
Yeah.
But there are 3 spots within it that if you change any one of them, you change the way it does the degradation, right? And so what this program is really good at is spotting, oh, this 150 amino acids tells me this is involved in protein degradation. And then it tries to pick out those 3 important ones to see if they're there or not. And so even if they're not there, it'll spit out, oh, it has the protease domain, but it's lacking what we call the active site. So the part of the enzyme that actually does the business. So these 3 amino acids that you can change, we call that an active site. And so you can look and you can see, okay, it's a protease, no active site. So we can predict that it's going to do the opposite of what it's supposed to do.
Gotcha. So the program itself, it's giving you the clue ahead of you needing to test it. So it's not that you're testing it and you're like, oh no, it's the opposite. It's that by looking at the prediction, you're able to figure out those differences. Yeah, and then know what you're testing for.
Yep. And so the student I was looking at it with, what she's actually working on, which I think is a really cool project right now, is she's looking at this protease that is predicted to degrade signaling peptides. And one of those is protein that's linked to Alzheimer's disease. And so what we want to do, and what's cool is, so this protease has about, I think it's like 8 or 10 different versions found in the venom of one of our wasp species.
Wow.
And so what she's going to do is synthetically produce each one of these proteases and test its ability to cut this peptide. And what we're able to do, because we have this sort of predictive protein, we know already where to look for the important sites. We know what it's predicted to be able to interact with. We can build a really good experiment because of everything that we know based on these prediction protein programs. But of course, it's a prediction, so we don't know yet. And so what she's going to be doing is making I think she has 20 on her list, but she's just starting her master's degree next year. So I don't think we're going to get through all 20, but we're trying to pick out a few really like high value ones to go after. And then she's going to make the protein and we're going to test, is it active as a protease, right? So we have some that have those 3 amino acids. So we think our hypothesis is that they're going to be active. We have some that are missing them. So our hypothesis is those are not going to be active. And then we have these predictions about what they should be able to cut. And so we're going to start testing those peptides as well, right? So we're going to look just is it active, yes or no? And then once we know whether it's active or not, we can look at, does it interact with the specific peptide that we're interested in?
And that presumably, that avenue, you can then also ask if there's off-target hits, right? So is it interacting with your peptide of interest, yes or no? And then, and or other stuff too, like is it just indiscriminate in what it's chopping up?
Exactly. So people first started studying this protein because it interacts with the Alzheimer's disease causing protein, but they very quickly realized that it interacts with a bunch of different signaling peptides, including ones that are important for heart function and ones that are important for other neurological functions and ones that hold our blood vessels together, right? And so you can't just manipulate this gene in a person because, like, you could cure their Alzheimer's disease by giving them heart failure, and that doesn't really help anyone, right? So it's what we're really interested in, and what the power of these venoms is these are, essentially lots of different variations of that one protein, and they have been selected over countless generations to do a very specific job, and so we think it's very cool and likely that we can, by studying all of these variations of the protein, understand how it selects which signaling peptides it's going to be able to cut, right? Like I would be shocked if all of these venoms do the same thing, and if they do the same thing that the human version does, right? Because our wasps, the context of them, right? They are in parasites. Parasites are using them to take over a host. You don't want to stop your host's heart if you're trying to parasitize it, if you're trying to live off of it, right? So we think they might be a little bit more subtle in their activity. And so what we want to be able to do is to really kind of close this gap of having all these different versions of the protein, learning what they all do, and then using that to essentially build a model that would let us predict how we could, let's say, synthesize a new version only does the thing we're in, right? Like ideally, wasp venom cures Alzheimer's disease and I get rich and we're all happy. Realistically, it's probably more going to be a case of, can we learn the principles about how this protein works? And then once we've done that, can we use those principles in a design way to come up with a better class of drug or a gene therapy that we could use that might protect against Alzheimer's.
So this is a good starting point, I guess. It's almost like in the typical drug discovery mode where you have a molecule, you test for something, or you have a molecule library, I guess. You find one of the molecules that does what you want, and then you change it a little bit to make it try to make it better, and then you keep tweaking, and eventually you have what is the best version of what you can make.
Yeah, exactly. But in this case, we've let evolution do all that hard work for us, and we're just going to test all the end products and see which one is closest to what we want, and then that becomes our starting point for the next round of optimizing.
Which is, it's brilliant that you're using naturally occurring things like in an animal system to treat human disease. Can you go into a little bit about that, Nate? Like, have y'all had any positive hits on that yet? Have y'all found a venom component that you could put onto human cells and help them in some way?
Yeah, so I can tell you about a few avenues that we're going down that are all kind of at different levels. So one of them is this Alzheimer's disease. So it's something that I've gotten really interested in. And we can express this Alzheimer's risk factor protein, the human version, we can express it in a fly. And when we do that, we found that it causes inflammation in their brain. That leads to degeneration of the brain, and they have cognitive defects.
Wow.
So really, just by putting the Alzheimer's risk factor into the fly, they get what looks like a fly version of Alzheimer's.
And is that at a young age as well? It's like you don't have to age them out. Like they hatch and they're already they're starting to degrade right away.
Yeah, exactly. So kind of they are, a fly's lifespan is measured in weeks. But they are several weeks older than look older than what they should be.
So like the molecular signatures of age, they're aged upon hatching.
Yeah, exactly.
that's by putting in just one gene. That's incredible.
One tiny little peptide is enough to do that.
From a human, the human risk factor, you put it into a bug and you give it a human style looking disease. Like that's amazing.
Yeah, it's really something. It's super cool. Something I really love about the fly system is we can do this. And we found a couple of our wasp venoms. We don't know the individual proteins yet, but at the level of whole venom, we've got a couple of them that prevent the inflammation response from happening in the first place. We're in the process now of trying to figure out whether it's doing it by degrading the protein, which is the student's project that I was just telling you about, or whether it's actually blocking the inflammatory response to the protein.
Gotcha. So in a nervous system specific way.
Yeah, exactly. Like, do we have a neuron anti-inflammatory protein that we could, and I don't really care which way it is because it sounds good either way, right?
No, it's cool either way, right? And presumably, like, if it's an anti-inflammatory on neurons, you might be able to use it for other diseases too that would cause like the same effect to it. Like, so naive question.
Yeah.
How do you deliver wasp venom to the brain of the fruit fly? And in particular, just that one molecule. Do you have a micro injector needle? How do you do that, Nate?
So we have a micro injector needle, but I'm not that good at using it. So what we do instead is kind of a multiple tiered approach, right? So the simplest, I say simplest because it's not at all easy. We can dissect the brain out of the fly. We can actually keep it alive in culture for hours to days.
Wow.
And we can then treat it with the protein and see what happens.
Okay, so you're like lobotomizing the fly, but all the neurons are still firing. It's the cells are alive.
You have a few days to play with it before it starts dying on its own. Kind of.
That's really clever. Just as an aside, that's a really cool technique.
We can pull it out and play with it. We can do it at the more molecular version of that, right? So we can say, oh, this is the protein that causes the problem, or this is the pathway that causes the problem. And instead of looking at the brain gross morphology, we can say, let's look at the activity of this protein in the right. So we can do that same kind of experiment, only instead of looking at the degeneration of the brain, we can look at expression of a particular inflammatory path. And we've been doing that, and we found a couple of pathways, inflammation pathways that seem to get turned on by disease. And so we can start using those as diagnosis almost of what's happening.
So if you know this pathway, let's say, is turned on in the disease state, if that pathway is blocked by wasp venom, then you know what it's kind of acting on, and you have some targets to focus in on.
Yeah, exactly. And then the final step. So once we've got to the point, with a student who's looking at the protease, right? Once we found or made what we think is the best protease to use, we will make a fly that naturally expresses it in its brain and see if that fly now becomes resistant to us adding the disease-causing human gene into the fly.
See, it's such an elegant puzzle that you're basically describing to us on how the system works, because we're understanding correctly, you think you've identified what the pathway is, so what component is being targeted by the wasp venom, and now you end up modifying that pathway in the actual animal and seeing now if it could be affected or not. So you're basically simulating the effect of the venom minus bathing the brain in venom juice.
Yep.
And seeing if you can get the same effect.
We don't have to do anything to the brain that could cause a problem. We just put that protein in, let it express it itself, and then see if it makes it resistant.
It's such an elegant system because you have the answer right there in front of you.
Yep, exactly. We just have to see what happens to that fly. Can it remember what it saw yesterday?
Yeah. So you can check, like you said, morphology, so structure of the brain, and that has similar consequences as in humans. You can look at the cognitive effects of it. You can look at, you were saying, how your student might be looking at the peptide and the protein, so you can look in a test tube on just a one-to-one molecular interaction. That's not just the basic drug discovery where a molecule does something that makes it glow. It's you're figuring out the entire each component and figuring out how those components are interacting and why that's important.
Yeah, exactly. We're trying to put together the whole mystery.
Yeah.
Like there's a lot to be said for let's just shotgun and find something that works. But by the same token, this has been such a hard road to go down, right? Trying to cure some of these diseases like Alzheimer's is kind of a famous example that is difficult to treat. It seems like maybe there isn't a shotgun approach that gets us there. And we actually need to understand step by step the biology of what happened so that we can come up with something that might be more beneficial to people.
Do you ever just sit there and think how incredible it is that you're treating or looking to treat Alzheimer's by looking at a wasp venom?
Yeah, I have days where I'm so busy that it doesn't even cross my mind what I'm doing. And then I have days where I come in my office and I sit down and I see I have 800 emails, so I close my email. And then I just take a minute and think like how lucky I am that I get to do something so cool every day. Right. And then I go out my office is sort of in the corner of the lab. So when I get frustrated by the emails or, the dean needs me to do whatever something I don't want to do, I turn off my computer and I wander out into the lab and I start talking to the students and see what they're doing and what they need help with or what data they want to share with me. And then I love my job again. And then I have enough activity to go back and answer those emails.
And Nate, we just got a raid from the lovely Lana Lux, game developer, with 300 people coming on in here to learn about SciAnts. Guys, welcome on in. Folks, all my people, please go follow Lana Lux, game developer working on the video game Strain here on Twitch. Also a lot of assets using the Unity game engine. Lana was on our Women in STEM month back in March. She is amazing. Please go drop her a follow. Raiders, welcome. My name's Balint. I'm one quarter of the stream. My wife, Lita, and I run the channel. We both have our PhDs in molecular biology, and we are having a very special SciAnts day today. We do SciAnts every day on Twitch, but today we've had two very special guests. We had Dr. Oded Rakavi earlier today. He was talking to us about his research on inheritance of memory and behavior across generations, the Dead Sea Scrolls and testing the genetics of the Dead Sea Scroll parchment, which is incredible, and using toxoplasmosa, which is a plasmodium disease affecting cats, use it as a mechanism of drug delivery. And now we have on Dr. Nathan T. Mortimer. Nate is researching, he's just telling us about using wasp venoms and venom components from wasp to treat diseases, including Alzheimer's. So these are the things that Nate is doing in his lab, and I was just asking him the question, like, how cool is it? Like, do you ever sit back and be like, I'm using a wasp venom component to try to treat Alzheimer's? And it was just like the mind-boggling coolness of that and how that system works. So everyone, welcome in. We've got some pinned information at the top of the chat about Nate's lab. Please ask questions. That's why we're here. We're all here to talk about SciAnts and a love of science. If that also sounds interesting to you, you can look back at today's episode. It'll be on YouTube, on our podcast, as well as just hit the heart button and you can have more science conversations coming your way. Nate, for the folks coming on in that are new, do you have any, you mentioned there were other disease states that you're trying to look at using wasp venoms. Could you give the new people coming on in here another example of like why wasp venoms are gonna cure me of my ailments?
Oh yeah, no, absolutely. So another one that I think is really cool is we've been looking at a fly version of leukemia.
Of leukemia?
So leukemia is sort of cancer of blood cells. What's interesting is there is a sort of a single mutation that happens in a really high percentage of leukemia patients. And it's a mutation that turns on a particular signaling pathway. And if you have this mutation, there's a very high likelihood of getting leukemia. And like I said, most people with certain forms of leukemia have this mutation. We have a fly that has the equivalent mutation in the equivalent gene, and it gets fly leukemia.
Wow.
So it makes way more blood cells than it's supposed to. It has the wasting. It has all of the same sort of impacts that we think about in leukemia. And I thought being a blood cancer, so blood cells are part of the immune system. And so I kind of thought, well, this might be a rich place to look for venoms that might have an activity against this disease.
And Nate, so could you put into context like why you were thinking that, just like from the fruit fly immune system and its dependency on blood cells? Like, why? What's like, because right, from stepping back, it's like, how does this gentleman get fly cancer and a wasp venom and blood all linked together?
Yeah, no, that's a great question. So thinking about it in its fullest context, right? So we talked earlier about this wasp, how it infects the fly. And when it infects the fly, it's laying an egg directly into the fly's body, right? And so flies have what we call open circulatory system. So that means all of their organs are just essentially bathed in their blood all of the time, right? And so this parasite is in the fly's blood. And flies will try to mount an immune response to kill the parasite. And they do that using their blood cells, right? So their blood cells are the cells that do the innate immune response. And so they're trying to kill off this wasp egg that's developing. And the wasp venoms are all evolved to block fly immune response, right? So I kind of had this thought, and the gene that we're interested in that's linked to leukemia, we've also shown that it gets activated as part of the immune, right? So it has, both in people and in flies, it has this important role in making immune cells and the function of those immune cells to fight off infection. But it's just when you pick up these mutations, it all goes wrong and it leads. And so sort of thinking this through of, well, this is a pathway that's important for immunity. The wasp has, say, 200 venom proteins that have evolved to block immunity. Maybe we have a connection, right? Maybe there's going to be something that targets this pathway specifically.
And you had mentioned earlier that you were working on multiple different species of wasp. And so a question from the chat is like, well, how do you know which wasps to start with, which venom components to start with? Because presumably they're all modifying the immune system right in different ways. How do you pick your candidate to go and try to treat this fly leukemia?
Yeah, that's an awesome question. And generally we don't pick, we let the data select for us, right? And so the first student to work on this project, she had this model of leukemia. She learned how to test it, right? So how to grow these flies and how to test whether or not they're showing the leukemia-like symptoms. And then she just started going through the list of all the wasps that we have, and one at a time infecting the flies to figure out which one changed it.
Wow. Okay. And that's using all of the cocktail, right? That's using every venom component, not yet exactly. Figuring out.
So if we just smack everything at it, what happens?
Right. It's like the dirty experiment, right? It's like you're just throwing everything at it, and if something works, then all right, we're good. We got something to run after.
Yeah, exactly. I think of science almost as like a big funnel, right? And so at the top, you're just going to try everything you can to see what's interesting, right? And so is there something in venom that does this? And that was the first student's experiment, and she found 2 species where the answer was yes, there was something in the venom that did it. And then from that point, you start narrowing down, right? So that's when you start looking for the individual protein or proteins that are responsible. And so that's what we're doing now. So this project has actually all been done by undergraduate researchers in my lab.
Wow.
And I have an undergraduate student right now who, what he's doing is dissecting the venom from this one wasp species that has the anti-leukemia effect we're interested in. And he's developed a way of testing the activity of the pathway, right? So he has set it up so that the blood cells, if they have the leukemia activity, they glow green when you shine a laser on them. And if you block that pathway, then there's no color.
Oh, that's so elegant.
Yeah, and so we have an instrument in the lab that can read thousands of cells at the same time and tell you how many of them are green and how bright the green is.
Wow, okay, so it's not just binary. It's a dial, right? So you can have that means you can make the protein better or worse eventually, right? Like, are you turning it down all the way, a little bit?
Yeah. And so he's developed this experimental setup, and a few other students helped, but they've developed this experimental setup. And what my student has been doing is dissecting the venom out of this wasp and then using different biochemical approaches to separate the venom proteins out from each other, right? So if you think about the venom as a cocktail of 200 proteins, right? But some of them are part of big protein complexes. Some of them are little individual proteins, right? So you can separate them by their size. And when you do that, you now have, instead of a cocktail of 200 proteins, you have, say, 10 cocktails of 20-ish protein, right? And so you can use their charge, their size, all these different biochemical properties to separate them out. And so he started with size, and he's been able to find one, we call it a fraction. So one fraction, one sub cocktail of venom protein that alone has the ability to block this leukemia causing pathway.
And Nate, it's not as simple, presumably, as killing blood cells.
No, the cells are still there.
Okay.
We have just been able to block this pathway's activity.
So that makes it even cooler, right? Because it's not just like, oh, you're just going to pop the extras off. It's you're actually acting on a molecular level. You're turning off them being bad.
Exactly.
That's amazing.
So they should still be able to do their normal jobs. They just have this one pathway is impaired.
So it's again going against the grain of that it's not a poison, right? Like where it's when we're trying to do chemo, we're killing off the healthy tissue, and we're killing off the bad tissue as well.
Exactly.
Here, you're just turning off that negative element of all the tissues.
Yeah.
And the animal presumably is fine.
Yeah.
So
I mean,
it's amazing. It's so cool.
Yeah.
That's and it's a wasp venom, Nate. Treating leukemia.
Yeah, it's super cool. And I actually have a friend in the department here who's a new assistant professor who his whole research is cancer, right? And he works with patient samples, and we've been talking to him about this. We have another anti-cancer venom that we've been working on, and he is so excited for us to figure out the protein because he wants to put it in cancer cells and see what happens. So hopefully within the next few months, we're going to have enough understanding of our system to be able to walk down the hallway to his lab and say, look, we've cured cancer.
And so is that next step presumably taking that fraction and further subsetting it so that you can get the smallest unit possible that functions and then figure out, okay, those one, maybe, hopefully, or two proteins, how do they do what they do?
Yeah, exactly. So the fraction that we found, we're pretty sure has a lot of proteins in it, unfortunately. What we found, and this is an entirely different project that I could spend hours talking about, but I'll try to limit myself. So when we look at the wasp venoms, it's this really complex cocktail. A lot of the proteins that they have, we would predict, right? So we can make predictions about how they work. We would predict they would work inside of the cell, but it's being secreted outside of the cell. And so one of the big questions is, well, how do they get in? And another one of the grad students in my lab has started answering that question. And what he's found is that the venom actually contains what we call vesicles. So you can think of it almost as a mini cell, right? So it's a little membrane enclosed chamber that carries protein. And it seems though these proteins these vesicles have a bunch of proteins in them. And we can stain the vesicle and we can watch them go into the host blood cells. And so what we need to do next is figure out all of the proteins that are found in this vesicle. And then we're going to have to then we can start winnowing down which ones are important for it once we can figure out what is in the vesicle.
It almost seems like the vesicle is interesting from a delivery mechanism, right? Of like that it has packaging something, finds the outside of the cell and just dumps it on in there. Like that on its own is also really cool.
Yeah, so that's actually why we got interested in it is thinking about delivery. And there are lots of strides being made in things like gene therapy, right? CRISPR guided gene therapy, proteins that we can put into disease cells that are going to help cure the disease. And one of the big bottlenecks is how do you get it to the right cell? And we kind of talked about this earlier of I'm not smarter than nature, right? Like evolution is a lot more likely to come to the right answer than I am sitting in my office thinking about it, right? And so rather than trying to devise a way to deliver the drugs or deliver the proteins or the genes or whatever it is, let's find something that already does it and figure out how, right? And so that's kind of what we've gotten into with these vesicles is we know that they carry protein. It seems as though there are different types that carry different proteins. They go to different cell types in the host. So one of the things we're working on is actually trying to tag them with different colors and then see where all the different colors go. So we can label them all the same, and they seem to interact differently with different cell types. But what we ultimately want to do is label each type separately so we can look at where they go and how they get in. Some of them seem to like actually pass into the cell as an intact little bundle of venom, and others seem to get to the surface and fuse and just release all the content.
Which, yeah, I mean, that on its own too, right? Like the differences in the kinetics of being released within a cell, like deeper at an organelle versus just globally right away, targeting to a particular site, like that could be revolutionary for therapeutics.
Yeah. So it's something we're super excited about. And my student has been making incredible progress figuring it out. He sadly is going to graduate in about a year. So I need to find the next super student who's going to take this on and work on it. But yeah, it's something that I'm just really, when we had the first indication that something like this might be happening, I was blown away and I was skeptical because it just seemed way too cool to be real. So we've sat down and I've had a couple of students work on this, and the most recent one is a PhD student, so he's been working on it for several years now. And yeah, everything he's figured out is just blowing my mind. Like, these vesicles all have different sizes, different charges. Like, there's so much we can do with them that is really, I think, taking us to something really cool, thinking about how we can deliver biological molecule, particular type organ diseases or whatever it's going to be.
I think it's a good problem to have, right? It's like, I am so mind blown because it is so cool.
Yeah, exactly.
Like, that's not necessarily the reaction that you normally have of like, what's going on here? It's like, wow, this is incredible. More, please.
Yeah.
Yeah,
exactly. No, it's the biggest problem that I have with projects is focusing on a small number of them. Right? Because for every project that I've told you about today, like, there have been like students working towards them and making good progress. But like, I know that we can't stay here for 6 hours for me to tell you about all the other projects. And another, 12 hours for all the random ideas that I come up with in the course of a day, right? So I think there's, it's a great problem to have, right? That if I had unlimited money and space, I would want to know what every one of these hundreds of venom proteins does. But as it is, we're focusing in on things that we think are really cool and potentially really important in terms of thinking about health and how we apply this to people's lives better.
From the chat, Nate, Sparky Pugwash says, it's okay, I have 12 hours.
Nice. I'm gonna need some dinner at some point, but I will filibuster.
Well,
I think we just need to lean on Balint here to invite me back sometime. I was mentioning ahead of the stream that you had mentioned the possibility that coming back, and I was like, I hope we don't scare him off before he's able to volunteer that again. But no, it's as always, Nate. Do we still have you for a little bit longer? Oh yeah. I haven't. My wife will text me when it's time to go home, and I haven't got a text yet, so we're safe.
And for you remember, we did have Alicia on the stream, but a few weeks ago, and we have to have her back on as well because we were already rabbit holing and like loving her research too. So it's a condition of the Mortimer family. We just have to keep having y'all back.
It must be. We talk too much.
No, it's just really exciting science, and that's the cool part is that this community of scientists and non-scientists in the chat, like there's just question upon question, and just people are just like, wow, it's just so cool, and like value what you're telling us, and like how it's a meaningful research and that strives that you're doing, like of how important it is that what you're doing, Nate. Like, it's really, really cool.
Yeah, no, it's and it's great, and I love this whole setup here that of what you're doing, right? This bringing people together and making science for everybody, right? Like something that I've done a lot. So you mentioned really briefly sort of leading this big genomics project. My favorite part of all of that is a lot of the instructors at these different institutions will invite me to give a guest lecture to their class. And if it's a school that has enough money, they fly me there and I get to hang out for real with the people. But a lot of times it's a Zoom call, right? And so I'm zooming into the classroom, and I've gotten to meet students from all over the country, people I would have never met. Like, I've never been to Oklahoma in my life, but I've given like 10 lectures to Oklahoma students at a school there, and I love these students because they ask me the most off-the-wall questions every single time. Like year after year, they have good questions, and so that's my favorite thing is getting like, they have to listen to me talk about the project for a few minutes, but then they get to ask all of their questions that they have. And yeah, like I often have to go back for a second visit because we didn't get to all of the questions.
Would you mind actually digging into a little bit, Nate, about this genomic project, like from your own mindset, like how it was, I guess, started, like what's your goal with it, like what's pushing you in this project, like why you're excited about it?
Yeah, no, so it's under an umbrella organization which is called the Genomics Education Partnership. You can look it up online. It's a really cool thing. There's a whole bunch of these different scientific projects going on, but they all use kind of the same like genomics framework. And so the goal behind it, we'll talk about the specifics in a minute, but kind of the goal behind the project is to bring research experiences to students who probably wouldn't get them otherwise. And so most of the schools that participate are what we call PUIs, primarily undergraduate institutions. So schools where there's not a whole lot of research on campus. And so for those students to get a research experience, which is so important, right? Like I wouldn't have done any of this if I didn't get to do research when I was an undergrad. Instead of making it where they have to have the ability to leave home for several months over the summer and get accepted into a program at some other school, which is kind of the typical way of doing it, but you lose a lot of people, right? There's a lot of students who are caregivers and they can't be away from home for 3 months at a time right? So how are those students ever going to get a chance to do research?
And presumably there's money involved too, right? Like if you're able to leave and like have an apartment or expenses, right? Just in general too.
Yeah. So it's a great model, but it's a really restrictive model, right? And there's definitely strides being made to make that summer research experience accessible to more people. But it's always going to be limited by money. And so there's a big movement that kind of is complementary to that of what we call CURES, or course-based undergraduate research. And so the idea is rather than make research something extra, something that students may need to sacrifice, something else that's important to them to be able to participate in, actually embed it in the classroom, right? So the students are going to class, learning whatever material they're learning from that class, but they're doing it in the context of participating in a real research environment rather than, I'm sure we've all done those. Anyone who's taken a science class before, you've done a titration where everyone knows what the titration point is supposed to be, and you feel like you just wasted half an hour doing the stupid
the droplets to get the color to turn. Yep.
Yeah, exactly. So that's not going to make anyone a scientist. Like, to be real, no one's going to become a scientist because they did that. But someone might become a scientist because they did a real research experience in their classroom. And so that's really what it's all about, kind of at the high level motivation is giving all those people who would otherwise do research, here's a research opportunity for you.
Gotcha. And it's highlighted around, what is the question highlighted on? So like you were saying, you're going to the labs either in person or on Zoom. So how is it connected to Dr. Mortimer here?
Yeah. So there's a couple of variations of the project. So the basic one is purely a genomics project, right? So we've kind of been talking about this, like, we've sequenced the genome. We've looked at the venom, so we know all the proteins. It's nowhere near as clean as what that sounds like, right? So when you do the sequencing, you have errors in your sequencing. You have sequences that don't map back to the genome. There's all sorts of mistakes, essentially, that happen along the way. And the computer algorithms are not great at correcting those mistakes themselves, right? Because they're the ones who made the mistake in the first place, right? Everything gets fed through these programs that try to assemble the genome, map all the proteins, predict all the proteins. And so what the students do is, one individual project is the student gets assigned what we think is a venom gene.
Okay.
And they have to go in, and we have, it's called the genome browser. So it's a website that has basically all of the genome or RNA or protein level information that we have about that gene in one place. And so the student student will go to this site and they learn beforehand all of the parts of the gene, how to recognize them, how to predict them. So they've kind of learned all about what a gene looks like and how a gene functions to produce a protein. And then they get into this data set and they look at their venom gene and they have to figure out, first question is it a gene? Like, is it a complete gene? A lot of the predictions that we have are like partial genes or partial protein. And so what they do is they go in, they look at all this data, they either confirm everything's fine, which very rarely happens, or they figure out what's wrong, like where the algorithm went wrong, and they use the data that's there plus the knowledge they've gained from class to actually correct it. And so the students in all of these classes are the ones who are producing, we call it a gene model, right? So these gene models that are actually correct. And so we've done a lot of work, looking at expressing individual proteins in the lab and all of this type of stuff. And all of that is driven by these students who are going in and using the genome data to make sure we're using the right model of the gene or the complete model for them. And so like students in my class, I think they do like 4 or 5 of these genes throughout the semester. And then, we talked a little bit earlier about the different programs you can use to predict what type of gene it is or what type of protein it is. And so then they learn how to use all of those predictive proteins and then they predict it. And so their final project is, well, I found this venom protein. This is its predicted function. This is an experiment that I would like to do to see how it works in a fly kind of thing. So it's a fun class. And I like those. Those are really great for these course-based research experiences because it doesn't require anything, right? All you need is access to an internet browser and obviously interest and excitement to learn it, but then you have everything that you need, right? It's very sort of self-contained. You don't have to pay a penny to be a part of this project. Then we have other students or other classes that are doing like wet lab, hands-on biochemistry or molecular biology. And those classes are actually making expression constructs for venom protein and molecular biology. And in biochemistry, they are expressing the proteins. Like we talked about earlier, they're testing their activity. So we have one of the venom proteins that we find in lots of different wasp species is a protein that looks like it's involved in sugar metabolism. And so we've had 4 or 5 different classes express different versions of this protein and actually test, do they break down sugars? And some of them are even figuring out how efficiently they break down sugars and comparing them to each other. And so these students in the classes are figuring out how individual venom proteins work. And what we want to do ultimately is be able to, we have this big cocktail. We want to know what the individual proteins do, and then we want to be able to put those back in the context of the whole cocktail. And so these students are doing really important experiments for us to be able to understand what these individual components of the venom are doing. And so it's really cool. And so there are classes at I mentioned Oklahoma, so Oklahoma Christian University is part of this. Siena College in New York is part of this. Wayne College in Tennessee. One of the UC schools that I'm blanking on, or Cal State schools that I'm blanking on, I apologize to anyone from there. But so there's sort of all the corners of the country and people all over getting involved in this. And most of these are students that have never done any research.
And it gives them real research experience because, like you said, it's not just an experiment that everyone does, rinse, repeat in every single biolab. It's you are discovering something new from the start.
Yeah. And so that's why I kind of go and talk about the project in their classrooms and, hopefully get them a little bit fired up about the research. And then what's great is a lot of times at the end of the semester, they invite me back and the students are doing their like final reports. They often do like a poster presentation about their project that they did. And so I get to like watch their poster presentations of what they found of their enzyme that they studied or their venom protein that they did the gene model for. It's really cool to be involved with the students and people who have never done it before are coming out with like, hey, I just figured out what this venom protein is. It's really kind of powerful to do in like 3 months or however long a class takes.
And does that that funnel, I can see it a system where, right, it funnels to each other, that you're making it in the computer only zone of like, here is the predicted function, and then you get to the other class that can do the wet bench stuff that can actually test its function, and then you could going to your lab again as a follow-up, like further mechanism. Like, it's almost a funnel again of discovery. Is that a fair assessment?
That's 100%, and that's why we kind of started setting it up this way so that we could have it, right? Like, it's super cool for the students to get to do it. They also really like the fact that they are collaborating with other students in other parts of the country.
Yeah.
Right. And I know that there are a couple of my collaborators on this who teach biochemistry classes, and they've actually been trying to figure out how to get their classes together to present to each other. So far, they haven't taught in the same terms. So that makes it really hard. Yeah. But I think one of them's schedule is changing. So I'm hoping this coming year they're actually going to be able to have a research symposium of students at 2 different universities who are working on the same project. And I'm trying to work on with one of them. I'm trying to get some grant funding. We won't talk about that process today, but I'm trying to get some grant funding. And one of the things that we want to get money for is that final step that you mentioned, right? Like, I would like for the student who has never done research outside the class, who has, like, let's say, expressed one of these enzymes and tested its activity, to be able to pay for them to come and visit my lab and do whatever is the next experiment that would follow on with their project, so they can just sort of see it to that next level. And obviously it requires someone who's going to be able to make that commitment of coming, traveling, and spending whatever it's going to be. But we want to be able to provide them housing and give them a paycheck so they can still be making money while they're doing something that hopefully is more fun than whatever else their summer job would have been.
Yeah, and that would be an incredible opportunity to take it from like the concept where it's just an idea, and then actually seeing it in a dish, like how it works and how it how it dysregulates and how it regulates in a beneficial way.
Yeah, I think that would be really, really great to be able to do that for some of these students who are starting with a piece of paper with an assignment and figuring out how to go through all of the, well, what is my gene, what does it do, and then they can come out here and we can make a fly that expresses it, and they can look at that fly's behavior or whatever it's going to be. I think would be a really cool way to kind of put the cherry on top of that experience.
And what's fun is the chat's already kind of hypothesizing on diseases that the sugar venom might work on. So is there any guess that it could be helpful for diabetes, for example?
Oh, that's a really good question. So we haven't dug that deep into it yet. The place that we've kind of started thinking about this is, and I have to get slightly technical, and I apologize to everyone, but when you think about glucose, right? So a molecule of glucose, which is a really simple sugar, goes into your cell. It can get metabolized, a bunch of different ways. We're only going to consider 2 of them, right? So there are different pathways that glucose can get shuttled down, and which way it goes depends on how it gets modified, right? And so one of these pathways produces energy, right? So it breaks sugar down to make energy. And I imagine most people who have taken a biology class have heard of glycolysis, so that's glycolysis, right? Taking glucose and making energy. The other pathway is kind of the opposite. It takes that glucose and uses it to create building blocks to make nucleotides for RNA, to make amino acids for new protein, to make lipids for new membranes.
So instead of like energy production, it's your building blocks. It's for the construction crew to have stuff to work with.
Yeah, exactly. It's giving the ability to make new cells or make new protein. And so these are very, very different uses of glucose. And what we find in everything from people to flies, in our immune cells, in our blood cells, when we get an infection, our body chooses to use glucose to make building blocks. And that's an important part of the immune response. If you mess with that ability, our immune cells don't work as well. Almost universally, pathogens want to go the energy route.
Yep.
Right. There's almost this fight for glucose and how it gets used. And we talked a little bit about malaria earlier, and something that's very cool in that context is one of the ways of diagnosing malaria is that the infected cells have really high levels of a metabolite from the glycolysis pathway. So from the energy producing pathway, one of the things that gets produced, people with malaria have a really high level of that. And it's cool. So And with the wasp, it's the same. So when wasp infects a fly, the fly ends up with really high levels of metabolite. And the sugar degrading enzyme that we're working on is part of this glycolysis path. And what's very cool is the students. I have a student who's just joined my lab this year. He officially joined in April. And he's going to be working on this project, and he's already made a really great start. And he's from Ghana. And before he came to grad school, he worked for the government for their version of the CDC, essentially, studying malaria. So he has publications looking at like genotype effects on malaria. And so he knows all of the stuff. When I mention the metabolite, he's like, oh, yeah, that's totally linked to this, and it's this pathway. And I'm like, yep, same.
That's awesome.
He's super fired up to work on it because, like we talked about before, it's really hard to do experiments in a lot of these sort of clinically relevant settings. But the same thing happens in our system where we can do whatever we want, right? And so that's something that we're going to be studying. And he's going to be taking the lead on that because he has this sort of personal experience with this disease. And studying this.
And that would be going from the idea that we can figure out how this works in a fruit fly, like all the ins and outs, like that, basically a giant web that we can draw out and how the system works. And then once we get it in this system where we can mess up any kind of factor and figure it out, we can then translate it, like to the mosquito, to malaria, or whatever else downstream that we want to compare it to.
Yeah, exactly right. Because we know observationally what happens, but we have no idea the relevance of it. We have no idea how we can manipulate it to make a difference, right? So just thinking about this like fight for glucose, right? There are two models that we have not been able to distinguish between. Hopefully we can in the fly, right? One of those is that the pathogen needs a lot of energy. And so it drives glucose down the energy production pathway. And so maybe we can target cellular energy as a way to boost immunity against these parasites, right? It could not care one bit about the energy, but it could really, its whole job could be to keep the glucose away from doing what the host immune cell wants it to do, right? Like, we talk a lot about, it's trying to block immune responses. Well, this metabolism is part of the immune response. And maybe by just converting the glucose to something else, you're just blocking an immune response, right? And so if we can see that the parasites don't actually care about the outcome, they just care about changing the signaling pathway in the host, that completely alters how we would think about treating it, right? Like in that case, targeting cellular energy is not going to do a single thing to help boost immunity against the parasite. You need to target the building blocks, right? You need to give more building blocks, right? So it's really important to understand, to kind of go from the observations that we have of the disease state and understand how they arise and kind of what the importance is in the interaction of those things happening. And then that changes how we can go back and treat them.
Yeah, it's like you were saying about with the Alzheimer's too, right? Where we've tried kind of the shotgun approach, but we might be more fruitful if you actually figure out how these steps work, right? Exactly. And same for like this venom. If they're just shuttling off the glucose and that's not what they care about, then you like altering those levels after the fact isn't necessarily what's going to matter, right? You have to like
exactly, like they're not going to care if you block their formation of whatever thing because they didn't need it in the first place.
No, that's it's such an elegant way of doing it, right? And figuring out then each step, and like it's not, like you said, it's not shotgun, but it's also very not just eye in the sky idea. Like there is a beautiful like pipeline that you've produced of like why I go here, why I go here, why I go to this step and this step, and it's like it's really cool that it also stems from undergrads at a completely different part of the country without any training to begin with that are feeding that pipeline to learn about how these components work and how we can help people.
It's really it's why I get so excited about being part of these big projects, right? Like it's not part of my job. I don't need to do it, but I don't think I could ever stop because it's part of this almost ecosystem, right? Where we're able to give these students opportunities they wouldn't have, and that opportunity is to make a really meaningful contribution to science that hopefully is going to help people combat infection. It's a system that is really important, I think, to be a part of and to try to promote like that. But yeah, it's a lot of fun. It takes a lot of time, and I've definitely had the advice from well-meaning people that I should drop it, but I I'm not good at listening to advice. So.
Wait, and I love the fact that you're not. And actually, that's a question from the chat that's directly connected to is what disease is currently, you think, the most promising target to be in some way treated with these venom-derived substances, like, or closest to being treated? Like, so it could still be a distance away, but like either really promising or you're just like really excited about it because this would be such a game changer.
Yeah. So certainly the Alzheimer's disease project is going to rank up there. I think that we are in the process of learning a lot more about how that risk factor leads to Alzheimer's disease, and at the same time, how to stop that. Right? Like, what are the funnel points where we can alter either that protein or the response to that protein that might have a big impact down the road? And Alzheimer's disease is obviously such a widespread disease that has such a devastating effect that people are very aware of the need to work on it. And so I think that's one where there's almost more opportunity than there is for a lot of other diseases, right? And so I think if we can make the meaningful contributions in understanding, I think there's going to be lots of opportunities for us to find partners that we work with to drive it to the next level, of go from what we understand to producing something that might help.
Right. So like those expertise would be presumably testing in the organism that is like a next level up higher, right? From all the concepts that we've learned in the fly to the synthesis to eventually presumably the human samples, right? Because you were mentioning some of the collaborators down the hall that have access to those specimens and the expertise.
Yeah, exactly.
Yeah. So that could, it's really, really cool. The ability to have this, it's just a really beautiful story of collaboration as well. Like it's a network that's building upon itself. You're not insular, just working at one lab in one university and that's not anywhere else. It's clearly a really beautiful connection between so many labs, expertise, and people.
Yeah. And that's something that I think is really important, that has really changed a lot in science over, say, the past 20, 30 years. And that is how important collaboration, right? Like there's always been a collaborative spirit, hopefully, among scientists. But I think with the technologies and everything that are being developed, we've kind of reached a point where you are unnecessarily limiting your ability to do science by not collaborating with, right? So like just within my little department here, and there's like 15 or 16 of us, there are people who can solve the structure of a protein, right? So something that we want to do when we find these venom proteins that are cool is actually determine the three-dimensional structure so we can understand better how they work, right? And I can't do that, but I can walk down the hallway with a test tube full of protein and say, hey, figure this out. And they can, right? There are people in the department who can do these. We talked a little bit about simulations. There's, someone in our department who can build a simulation of a cell, and he's interested in how mechanical forces impact cells. And so he can do things like, oh, well, if we change the angle of a protein in the membrane of a cell, how does it alter the cell's function? And like that just blows my mind. I don't even have a concept of how you would start doing that. Right. But the fact that we have that kind of expertise and he's the one who's been helping us with a lot of our predictions about the venom protein. Right. But like with these vesicles, he's like, oh, yeah, once you figure out what they're made of, I can help you simulate how they interact with the cell. And like, I would never in a million years be able to do that. I probably wouldn't even have thought that that's something that we could do. And he heard this story and was like, oh yeah, just, like once you figure it out, let me know. And we'll get a student and we'll figure out how to do this. Right. And so this is just a group of like 15 or 16 people. And there's all this crazy stuff available. And like with me coming here, there's now like, I provide a different angle for all of those people. Right. So one of the people who's, a world leading expert in studying a particular class of protein, they can understand them structurally. They understand their interactions. They have no idea if it matters in a cell. And so I'm going to help her students get these proteins that form different structures, put them in fly cells, and see how it changes the function of that cell. Right. And that's something that they can't do. That to me is like dead easy. Like I came up with a plan in like 30 minutes and, to them is impossible. Whereas like I have no idea how I would figure out the structure of a protein. And they're just like, here's our list. Just do these things. Right. so bringing together all these different people with such different backgrounds and personal backgrounds, like scientific trainings and what they're interested in and what skills they can do is really, that's where science is going, right? Like my lab doing what my lab can do only gets us so far. But as soon as we talk to the lady down the hall, we're all of a sudden opening up a whole new world of what we can do. And it's just really, it makes it a lot of fun. And I think that's the way science has really changed, is it's not just like, oh, cool, I have a collaborator. It's like, okay, I want to answer an important question. Here's the collaborators I need to find. And, working at a school like this, an apartment like this, a lot of them are on my floor. So that makes it a lot easier, right? But we also have people in New York that we're collaborating with, Oklahoma that we're collaborating with, right? Like all over the place. And everyone has their own skill set that they're bringing, making a contribution to a project that wouldn't happen at all or would be kind of crappy without their input, right?
And it's like you're making it better with all those niche expertise. It's not like you're trying to learn all of it. It's like, look, I don't know this, and that's okay. I'm going to get you, the expert, to do that part of it.
Yeah, exactly. Like, I would never be able to do my job and learn how to do all these other techniques at the same time.
Or right? Any of your students too, right? Like there's no way that you can have them versed in all of these things.
Go off and learn how to do structure determination on their own. It takes that interaction between people. Like we have a paper, so this vesicle paper, we're hopefully going to be able to submit it soon. We're working on the last experiment right now, but we have a collaborator in that project who runs the electron microscopy facility. And so she's the one who figured out the technique we can use to literally look in the venom and see that these vesicles are there. And then we have a collaborator from Illinois who's a chemist, and he's a chemist who studies nanoparticles, and he knows how to take the solution that contains nanoparticles and count them and see how big they are. So we sent him some venom, and he did that. And we have another collaborator at down in Alabama who is an expert in, we talked about this mass spec to identify proteins. He runs their whole facility there. He's about to retire, lucky guy, but he runs their whole facility there. And so we sent him, hey, we think there's a vesicle here, like what proteins are in it? And he told us, right? And so it's kind of like this paper that comes from my lab, but we have 3 like experts in different places that are unrelated to my lab that have made these really, really big contributions. And it feels almost like our job has just been to hold it all together and like find the thread that runs through what all these other experts are doing, and how do we tell the story that's forming out of all these people doing vastly different things, and how do we put it all together and make sense of it. So it's really, I love working with people who are way better at me than things at things that I don't know how to do.
I mean, that's not an attitude that everyone even shares today, right? Like that's a very like, you have to be, I feel, humble and wanting to work, and what you don't care about it's not about the credit, right? It's about coming up with the answer, and you just want to do it the best possible way you can.
And you certainly run into people who the credit is the important thing, so they're willing to let you do all the work and then they do the last piece and take credit for it. And that certainly is part of how science works, unfortunately. But I've gotten better with time at being able to talk with people and get a sense of what their motivations are and be, choosy about who I'm going to collaborate with. If someone gives me bad vibes, that's the end.
Which is a skill on its own, right? Because it's especially like when you start out, it's just exciting to be, you're almost the new shiny toy. Like, oh, this guy does wasp venoms. Like, let me hang out with him. And it's like, oh, how can I use this to my own advantage versus like how can we just further knowledge overall?
Right. Yeah. Like that's something. So like my friend Dan, who's the guy who is the cancer biologist, like he could very easily have said, oh, give me some venom and I'll figure this out. But instead he's like, oh, we can work together and we can test cancer, we can test patient samples, right? It's a very similar idea, but the way you frame it says who you are,
right?
Like, oh, I can exploit you versus, hey, let's do this thing together, right? And it's the same thing in the end of what's going to happen technically, but it's such a different just mindset going into it that it's very, once you get attuned to it, you start to get really good at, okay, this person wants to actually work together and this person's going to screw me over the first chance they get. And it's a hard lesson to learn, and I've learned it, probably more times than I should have. But yeah, it's an important kind of radar to have when you're trying to collaborate with people. For sure.
We had a point from the chat about a Nate deep cut story that I've told about a weird collaboration. Do you remember when we got an antibody from a lab and we kept testing it and it kept not working and it ended up being water?
Yes.
Those are good times.
So there you go. Yeah, there's a good example of not willing to collaborate.
Oh, and just for the chat, you can't tell by eye that it's not antibody. Like, it is the same clear liquid as by eye as water. And so we're testing and dissecting and it's not working. And finally we put it on the, what did we, like on the nano?
Yeah, I think we just did like a spectrophotometer reading. And so this is an instrument where it uses absorbance of different wavelengths of light to tell you what's there. And so like protein, which an antibody is made of, is just type protein, has a very distinct signature, and DNA has its own signature, and lipids and sugars. And so we put it on and we read it, and there was no signature. So I don't even think they sent us buffer. Like, they just sent us water. Like, there was no chemicals in there whatsoever.
It's very clean. Very, very clean.
They just opened up that tube and like held it under the tap and like, screw those guys.
So yeah, those we now know to avoid.
Yeah. So they are on the blacklist forever.
So Nate, I don't know if we have time for this because I don't know if Alicia's texted you that you need to go.
It's probably getting close. Yeah.
So maybe we can hold this question for next time. But there was a question about, you were talking about like autoimmunity and recognition of self and non-self, and that was one of your, I guess, even harkening back to the TV show in Canada from Dr. Suzuki. Yeah. Probably we don't have time for it today, but folks would like to hear a little bit about that journey and how you model an autoimmune disorder in a fruit fly, and what is that? How does that connect back to the venoms and everything else of your interests? Yeah,
well, I'll tell you what. Let's do a short version of the story, and then hopefully it's good enough that you want me back, and then I'll give you the long version of the
Perfect.
story.
Kat, are y'all good with that? I think we're good with that.
Yeah. So hopefully, I mean, if anyone wants to kick me out, go for it. I've been here for a while, but I'm happy to go through one more. So this whole idea of
Nate, the chat is saying we already want you back. So before you left, we want you back.
Good. Excellent. Thank you. I will definitely see you again. So I was really interested in looking at this self versus non-self recognition, and in particular the self-recognition, self-tolerance, right? So the ability of an immune cell to know that this is my own tissue, don't mess with it, right? That seems like such a simple thing, but we never really think about it when we're thinking about immunity, and it's really, really important, right? So people tend to think of immune cells as, they have to be able to find the pathogen and kill it, which is important. But at the same time, you have a bunch of cells running around killing stuff. You want to make sure that they don't do anything to you too. And so there's a whole other system for that. And this is one of those examples that I was talking about earlier, how most of what we know about human immunology is focused on acquired immunity, right? And so for decades, everyone has tried to describe every autoimmune disorder as being an acquired immune. And it's very obvious that that's not true, because we don't understand how most of them work, for one thing. And they don't seem to be linked to specific, acquired responses. So it seems though innate immunity is important. But like we were talking about, these two branches are so intertwined in a human immune system or a mammalian immune system that you can't really pick them apart very well. And so that's something that's really, I thought, well, a fly might be a good place to look for that. And I was reading old papers. That's always a good way to get inspiration, right? Is something that someone 40 years ago thought was really cool, and no one's figured out how it works since. Like, that's a really great thing to go back and like pick up on and work on, right? So this is one of those papers. I think the very first paper was 1969, and until we started working on it, the most recent paper was like 1982, something like that. And and then there was a big gap after that, and it's because they found this mutation where the fly's immune system responds against the fly's own tissue as if it was a parasite, right? So the response that they do to that wasp egg, they were all of a sudden doing to their own tissue, and it was only one specific tissue, so it's a really weird thing.
So that must mean, just like by eye, there's something okay about all your other tissue, but there's something wrong with just a subset of it. So you've got like a built-in control almost, like as you're looking at this animal, like this is okay, this is not, what's the difference?
Yeah, exactly. And so just sort of thinking about it and looking at what was happening, it very quickly dawned on me that this might be the way to study self-tolerance, self-recognition in an innate, because to me, looking at it, I couldn't think of a better explanation for what was happening, right? That these immune cells are all riled up and they're attacking one tissue in the body and no other part. So that one tissue must have be missing something that's saying, hey, don't attack me. And so we wanted to get into it and look at it. And of course, very quickly realized that there's a reason why no one had worked on it for almost 40 years, 30 years at that point, because it was really, really hard to figure out. And it turned out to not be just one mutation, but it was two mutations, but they were in genes that were so close to each other in the genome that it was really hard to tell them apart. But if you just changed one or the other, nothing happened. And so it was this whole, we can talk about genetics next time if anyone wants to lecture on genetics, I'm more than happy. But
I think people would love that, Nate. We're a big fan of genetics here. Yeah.
Okay, yeah. We'll come back. We'll talk about the pure genetics of all these crazy experiments that we had to do.
Because of the teaser, you actually did like classical genetic mapping to figure out where these are located, in addition to like more modern techniques as well. So it's like, that's why I love that story so much, because it's such a beautiful journey from the 60s to modern times to figuring out how this works within an animal that you have a control built into it.
So we were doing the techniques that were first done more than 100 years ago alongside using the latest sequencing technology, and we would not have figured out what was going on without doing both of them at the same time. It was an incredible amount of work for one sentence of the paper. But we were able to figure out that there's a mutation in this gene, and it encodes a protein whose job it is to modify other proteins. And it attaches, sugar seems to be a recurring theme, it attaches a sugar group onto protein. And that's something that happens, a lot in our cells. Not so much within the cell, but a lot of the proteins on the surface of the cell, or proteins that get secreted like into the blood, a lot of those have sugar groups attached. And those sugar groups do a whole bunch of different jobs, including helping proteins bind to each other, to recognize each other, to be stable, to fold properly, right? They have three-dimensional structures. And so this risk factor, right, for the autoimmune response, its job is to put these sugar groups onto other proteins. And so we're at this point where we think that there's something in the immune cell that is recognizing a specific sugar modification that's found at the surface of the tissue. And that if you get rid of the sugar modification, the cell no longer sees it as, this is me. And so I have a student right now an undergraduate student right now, who has, we think, genetically figured out what that receptor is in the immune cell that is seeing the signal. And when he gets back from he got accepted into a pre-med leadership program in Pennsylvania or somewhere. He's on the other side of the country anyway for a couple of months. But when he gets back, we are going to try to test the biochemistry. We're going to see if we can directly bind this receptor that we think is doing the signaling with tissue that has or does not have the proper sugar modification.
Ooh, okay.
To see if that is how it's sensing self versus non-self.
Nice. And that again could be the mechanism, or a mechanism by which autoimmune disorders are happening, and a member of chat just said that an impression is that cells could always be theoretically under attack from neighbors. It's just it's that they're passing the quote unquote tests. And so you're figuring out what are the tests that are being examined on each of these cells and piecing that, like almost working backwards on that front to make it work. Yeah.
Yeah, that's 100% the right way to think, right? So there are immune cells, we call them surveillance cells, right? And they are all the time testing everything they encounter. Is this me? Is this me? And if the answer is ever no, that triggers an immune response. And as long as the answer is yes, then everything is cool. Right? So these cells now no longer have the yes. Right.
So now it's like, well, what is that? Yes. Like, what is that? How do I genetically make it?
We're trying to figure out how that system works. And what's really cool is when we look, so again, sort of thinking about the observations that we have clinically and how that relates to research, right? We know people with autoimmune disorders, and most of the work has been done on lupus, but other related autoimmune disorders too.
We actually did a big fundraiser, Nate, in May for the Lupus Foundation of America.
Oh, fantastic!
And that's something that we actually had on, and we were talking about lupus and the autoimmune component, and the self, and that's actually where we've talked to the community about self and non-self.
Oh, very cool!
It's all due to in that context of a charity. So it's really cool that you're bringing it back to lupus.
So that's one of the things. So people with lupus have altered levels of protein sugar modification. No one knows what that means, but they're messed up. The student who has found the receptor, so the receptor belongs to a class of receptors called integrins. So these are receptors on immune cells that they use to interact with your own tissues. The human version of this protein is the key risk factor for lupus, and it's the only risk factor that's been found in every population of people who have lupus, right? So in America, Europe, Africa, Asia, wherever people do a study looking for lupus risk factors, they find the human version of this protein, and that's the one that seems to be what we're studying in the fly.
And Nate, we actually just got rated by Streaming Falcon, who is actually a real-life psychologist here on Twitch and does gaming content. One of our really, really good friends, he said he didn't make a raid message because he wanted to respectfully raid so we don't interrupt you. I decided to interrupt you anyway. Guys, welcome on in. Actually, Falcon's been here earlier today too. He was listening to our earlier interviews and the start of yours. So he's brought over his community as well to be able to meet you and and learn about the incredible science that you're doing. For the raiders coming in, we are wrapping up with Nate because this kind sir does need to go home and have dinner. But we have two potentially fast questions, Nate. So one is there. So for those coming in on the raid, Nate was my mentor when I was an undergrad. He was also the best man at my wedding. I text this gentleman on the regular. There was a question if you have any silly, embarrassing Balint stories that you'd be wanting to share.
Oh, no, of course not. Balint is 100% the serious gentleman at all times. I almost made it through the entire sentence. Let's use that one as a teaser for inviting me back. How about that?
Perfect. So next time you'll tell us.
Next time I'll dish some dirt.
And then I guess just like a really, really big component of honest to God, like why you're even here today. Like why do you think doing something like this, going on to Twitch and talking to non-experts, or even like designing a course for undergrads that have no training in venomics and teaching them how to identify new venom proteins and what they do and what they function as a whole, why do you think this is important? Why is it important to engage with the general public about the sciences, and why do you, Nate, do this?
Yeah, no, that's a really good question. And I will give you the short version, but there is a longer version too, most likely. So for me, growing up, so I'm from Canada, born in a little farming community, and no one in my family's ever been to college, right? I have like one great uncle 8 times removed who was like a doctor back in England. Like that's the extent of education in my family, right? And I knew it was something that I wanted to do. And I always loved science, right? So we talked earlier growing up, watching on TV, seeing real scientists talk about science and how motivating that was. I had no concept when I was starting out that I could do it, right? Like it seems like there's a class of super people who are the ones who are become scientists and the rest of us get real jobs or whatever, right? And so I think a big part of it for me is being able to tell people who are like me that you can still be like me. Like, it doesn't matter who you are, where you're from, that science is for you too. And whether that's your career or your hobby or whatever it is, there's no barrier that really prevents you from doing. There certainly are a lot that get put up, and a lot of them in our own minds that we put up, but realistically, science is for everyone. I love coming and talking to people who are maybe not professionally trained scientists who love science, right? Because it's way more fun than a lot of the people I talk to on a regular basis. But I just, I love the fact that there are people who have different career paths that think what we do is interesting and want to know more about it, right? And maybe, like, to them it's a hobby, and then they talk about it with their kid, and their kid becomes a PhD student in my lab in 10 years' time, right? So I think a lot of it is just making science visible, but making it visible in a human way, right? Like, I'm just a guy. There's nothing special about me. I just really like science, and I put in some work, and I found myself here with this really cool job. And there's nothing really that would stop anyone else from doing the same path, or from encouraging their kids to go down the same path, or their grandkids, or whoever it is in your life that is making decisions about things like that, right? And I don't think that's a message that ever gets sent out, right? I think there are too many people who reach like spokesman status for science or for any other type of job who see themselves more as a gatekeeper, right? Like, I'm here because I am special. I got a PhD because I am special, not like I worked really hard and I did this thing and you can't do, right? And I think that's what the real message needs to be, is like, whoever you are, yeah, if you're excited by it and you're willing to put some work in for it, you can be a scientist, right? And you can do that through getting specific training. You can do that by coming to events like this and learning something new, and going and checking out some resources and getting involved in a citizen science project, right? It's not something reserved for other people, and that's why it's so important to me.
Nate, I can't think of a more beautiful place to end today, but also to tease future come-ons. Thank you for that really beautiful answer. I think what's really been fun today is both of our interviews today, both you and Oded, echo that exact same message, that the scientist is not made by the opportunities that you're given, like that makes you special about, like, oh, just because you got to go to this school, but it's you as an individual, it's your brain, it's your curiosity that makes you that scientist. And thank you for further reaffirming that to everyone in the community. We have, like I said, a huge diversity of people who listen, who watch, in all avenues of life, who just love science. And I love that your message is, you can do that.
Yeah. I mean, if you're here listening and enjoying it, you've done the hard part, right? Like, that's what it takes to be able to do science. Just that love, that willing to get outside your own box a little bit and to think in different ways and be creative and be willing to be wrong half the time, at least. That's what a scientist is. There's nothing special outside of
Nate. You are extremely special to me, as you're my best friend. But thank you very much, sir, for your time today, for your expertise, for sharing your stories with our community. We can't wait to have you back. Please have a lovely rest of your evening, and whenever you want to come back, it's an open door, sir.
Okay, excellent. That sounds great. I will. We'll definitely cook something up.
Thank you very much, Nate. Have a wonderful rest of your day.
Yeah, awesome. Thank you. Thanks everyone for coming and listening to my nonsense for however long I've been here for.
Well, we're getting a bunch of heart emojis and thank yous for even first-time chatters who've never said anything before coming out and saying thank you for your time.
Oh, excellent. Well, excellent. I'm glad people enjoyed it, and make sure keep in touch with this guy and his stream, and then hopefully I'll see you all again next time.
Thank you, Nate. Till next time, sir.
Yeah, awesome. Bye. Thanks, everyone.
Bye, Nate.